Right, tēnā koutou e te whānau. How's that all coming through?
Yep, we can see it.
Great, okay. No whakatua hau e mahi ana ahau mō te papa atawhai, called Josh Kemp tōku ingoa. Hi everyone, I'm Josh Kemp from DOC, based in Nelson, and with help from my team, I've been compiling and analysing tracking tunnels data from right across the country for more than 15 years. We crunch the numbers on percent kills of rats and mice and look at what makes for a good kill. Today I'm talking about rat kills. We've got two key messages expressed here as little rhymes that we hope will stick in your ear. The first one, rat populations in growth phase resist our bait and make us pay, which is about rat populations growing, not rat individuals. It's not about adolescent rats being hard to control because they're having a growth spurt. It's about populations. The second rhyme, the same dope used repeatedly will put your rat kills all at sea, which is about selection for genes that assist survival of 1080 drops. I've got 3 lines of evidence to support these messages. I've got a new analysis of tracking tunnel data, which is actually bulk of this talk. Have a little look at what previous authors thought. Mostly they agree with the kind of thing I'm saying. And the third is that these rhymes are just plain common sense. So let's start on the tracking tunnel data. We have data from 105 aerial operations over 15 years where tracking tunnel lines have been surveyed before and after the 1080 drops at a suitable proximity to the drop itself. Over 1,200 tracking tunnel transits in all across those 105. And plotting the percent kills achieved, 79 of the 105 have reduced the tracking rate by 97% or more, which is my red dotted line across the top there. And where operations have missed this target, the conservation goals for species at that site will very likely have also been missed or delayed. Or even worse, an emergency rat control operation had to be done in the form of another 1080 operation. And these operations cost a lot of money and there's a lot riding on them. The worse they work, the sooner we'll have to come back, diverting money from other sites and people's time from other conservation priorities. And the worse they work, the less confidence we have in them and others have in us. Plus, poor rodent kills are likely to mean poorer stoat kills by secondary poisoning, meaning we have to pay more for stoat trapping at sites where stoats are a primary limiting factor. So they make us pay. We need to do better than this. We need to get more of these operations up over that red line. We aim to improve things a little bit, hopefully, using a big data kind of approach to explore what things from a bewildering array of possibilities have affected kill rates. The blue things are design and engineering things up on the top left, whether to pre-feed, what size baits, how many baits, how to sow them, etc. There's lots of these factors and they all get decided in advance. The green things on the bottom left are weather related, a number of fine nights after baiting, how soon poison follows pre-feeding, how windy it really is up at 5,000 feet when the helicopter is over the range out of sight. We try our best to control these things, but in the end, it's not entirely up to us. The red things on the bottom right are nature things. How many other bait consumers are about? Possums, mice, deer. How much natural food is there? What is it and where is it? Which stands to affect rat foraging and ranging behaviour. These things are largely out of our control, but sometimes we can roughly gauge them. Then on the top right, there's history, treatment history of the site. Are there bait-shy animals persisting from last time? Have resistant behaviours and physiological tolerance proliferated under the strong selection pressure we're imposing? It's a bit like a school or a university with a Department of Engineering, a Department of Meteorology, a Department of Biology, and a Department of History. I think it's fair to say we've invested more in the Departments of Engineering and Meteorology than we have in Biology and History. It actually seems we haven't come that far at all. In the engineering department, we've messed about with sowing rates. This plot's got nothing to do with kill rates. It's just to show the changes that we've made in sowing rates over time. Each dot represents an operation sized by the number of transits that we use to measure rat kills. Sowing rates up the side, and we've mostly done about 167 pellets per hectare. That's this line across here. Which equates to 2 kilos per hectare of 12 gram pellets or 1 kilo of 6 gram pellets if you're used to talking about kilos per hectare. I prefer to, or we've done this analysis in terms of sowing pellets per hectare just to get that bait size thing accounted for, because we think it's all to do with baits in home ranges, not the size of the bait. But lately, you can see we've upped these sowing rates. So again, we went right down here to quite low sowing rates, and we've upped these sowing rates again, particularly in 2019, in an attempt to engineer improved rat kills in the light of some of the poorer ones that had occurred. We've also messed about with flight path spacings. This is the meters between the helicopter flight paths. There's a general kind of up and over pattern here. We got pretty loose in about 2014 with some very wide flight path spacings, and then we've clawed it back in 2019, culminating in this series of what I call kind of over-engineered operations, intentionally over-engineered, so that we knew if they didn't work, we could rule out engineering and gaps, particularly as a cause of failure. From the Department of Biology, we've been doing lots of operations at times of rapid population growth. So we've got population growth on the side. The black line is a zero growth population. And the dots again are operations, and if they happened above the black line, that means the population was growing at the time. Below the black line means they were going down. And anything above 0.1, which is up here, means the population is skyrocketing, as happens in big mast years. And you can see, you can pick out the mast years here. 2019, lots of operations and a growing population. 2016, and then you can also pick out the post-mast years, which are when operations have happened in declining populations. And usually these operations were intended to happen in the mast year, but got delayed by weather or demand for helicopters, and got postponed into the post-mast year. So we've been doing more and more of this, trying to control rats while the population is growing. From the history department, and here each dot is a tracking tunnel transect, just so you can view all 1,200 of them at once. A few of our transects are up to 8 treatments now, and we're still adding new hectares in 2020, but we're up to a full spread of first-time treatments right up to 8-time treatments. And by definition, because we keep going back to sites, there's a general increase on average in the number of times the transect's been treated. So has any of this actually affected our rat kills? How do you answer this question? Trying to solve it with the one-night tracking tunnel survey database was my challenge. The human brain simply can't cope with more than a couple of variables at once, at the same time as allowing for the different sample sizes and different operations, but your computer can. What I needed was a big, ugly statistical model like this. These are the variables where we had enough data for us to actually model their effect on kills. 4 from the engineering department, 2 from meteorology and biology each, and 1 from history. Treatment number. And treatment number is just a very rough thing. It's just like how many operations has this transect had before. And they're not all the operations are not the same. So the intervals between treatments and the sources of repopulating rats between treatments aren't all the same. So it's just a very sort of crude first go at quantifying an effective treatment history. And we modeled 4 interactions between growth and pre-feed. Maybe pre-feed doesn't matter so much when rats are hungry. Maybe sowing rate only matters when home ranges are small, when there's billions of rats. The modern statistical game is to run hundreds of models comprising all possible combinations of these variables. And after waiting for hours, you get to see which ones matter and which ones don't. So I'm not going to show you all the statistical stuff. I'm just going to give you a list and show you which ones didn't matter first. Bait size, the number of fine nights after baiting, and the number of rats at the time of baiting, and the interaction between rats and sowing rate were all basically completely unsupported. So we can put those aside. We don't need to worry too much about those anymore and simplify. Now, it's a bit better down to a few less variables. Now, sowing rate and flight path spacing were quite confounded in the data set, mostly due to that over-engineering that we did, where we increased sowing rates and flight path spacings at the same time in 2019. And so it's a bit hard to tease them out, and we're not too surprised to see statistical support was kind of mutually exclusive. You can have one or the other, but not both at the same time. And you don't really know which one's making the difference because they're kind of expressing the same thing. The essence of these two is that they reduce gaps in coverage. So let's just combine those into one and call it gaps. So here we go. This is what we've left with: pre-feed, gaps, pre-feed interval. That's the days between pre-baiting and toxic baiting, the growth rate on the day, the treatment number, and a couple of interactions. Growth mattered more at longer pre-feed intervals, and gaps mattered more at higher growth rates. And support for these two interactions wasn't terribly strong, but it was there. So these things are supported. But does it matter? To see the difference they made, we plotted predictions from the best model, putting aside treatment number for now, with pre-feed interval along the bottom and moving from a crashing population up on this top chart down to a skyrocketing one at the bottom with different attention to gaps, in this case sowing rate shown by the different colours. Blue meaning we were up to 3 kilos per hectare, and red meaning we were down at 1. And you can see that, well, basically it means that we only need meteorologists when we're working against biology, and extra engineering only gets you so far against biology and unkind weather. If this is making you actually fancy using more bait or extra overlaps on swaths to combat the downside of baiting when there's lots of food, actually the second best model is quite competitive, where it boils it all down just to growth and pre-feed interval being the primary determinants, suggesting that what we did in 2019 with the over-engineered gap elimination had marginal effect at great cost. Our basic engineering is fine, and if we do 1080 when growth rates are naturally moderate or low, it should be cheap and reliable. But baiting while populations are growing is quite risky. What about that treatment number effect? Well, after controlling for all those other variables, the tracking tunnels are giving us a stern warning in my view. Hopefully this speaks for itself as we move across from first-time treatments, third-time treatments, sixth-time treatments. You can see by the time you're getting into sixth-time treatments with growing populations, these things are compounding to actually make it look like it's just not a very good idea. Whereas your things are looking much more reliable, and perhaps some of these other things start to matter less if you're doing your operations when rat populations are actually starting to decline themselves. And it's not such a stupid idea doing these things when they're starting to decline. You might argue that we should just let them decline naturally, but actually there's a lot of damage done to our wildlife during those decline phases. Right, so there's my key messages: rat populations in growth phase and the same dope used repeatedly. Turning to my second line of evidence, just reading back through the literature on this stuff, I found these messages were hinted at repeatedly. Craig Gillies found rats took more bait in winter. That kind of fits. Graham Nugent found that highly engineered sowing rates and sowing patterns made no difference. That fits. John Innes and co linked repeated use of the same poison with poor kills, as of many overseas studies using other poisons. And my final line of evidence, they're just common sense. Which isn't very scientific evidence, but nonetheless, I think it's good to remember. When I practiced this talk on my wife and kids, they were like, "No, that's just common sense." Now, I had to admit that my job's not rocket science after all. It can be more about dispelling myths and legends and breaking old habits that formed as accidents of history. Yeah, so what changes? Well, first, I think methods of overnight tracking is awesome, and I think we should still do a lot of it, but actually, I think we'd benefit from a more sensitive measure after aerial 1080 when rats are actually very scarce. It's quite hard to pick up the kind of subtle differences we're getting once you're pre-feeding with one night tracking tunnel surveys. So we should have a bit of a think about that. I think we should angst much less over bait size, sowing rate, number of fine nights and flight path spacings. There's a lot of angst over that in the department, and I think it'll save many hours and lots of dosh if we don't worry about it. Instead, think hard about operational timings, how to avoid baiting and growth periods in our forests. And for eradication operations, like maybe if we get around to Rakiura, Stewart Island, or for ZIP doing operations in the West Coast, remove forests or anywhere, or even in fenced sanctuaries, anywhere, or islands, you're trying to eradicate something, it's going to work better if you manage to time it for a low or declining population phase. And finally, let's proactively study population level resistance and keep our toolbox a little bit diverse so that we can change up the baiting recipe now and then and keep these things working as long as possible. Kia ora.
We're back on track, folks, so we can have some questions for Josh, but there are no questions yet, which means I have to make one up, which is tricky because I talk to Josh all the time. So I suppose my question is, Josh, what I think you've already said it, but say it again. What do you think the main new research lines that this will lead us down?
Well, I think for a start, in terms of, I think this whole idea needs to be needs to be tested more. I think we need to think about, particularly in these mixed beech forests and in upland beech forests, going into mast events with already elevated rats because the intermast period's been warm, that we need to have a go at trying to work out whether or not we can get the outcomes we want for the birds and bats and co, with operations that are either before or after the seed period, the growth period, or both in some cases. And I think in the mixed beech forest, we're just going to have to face the facts that one pulse of control is not going to get these species through the kind of rat events that we're seeing in those forests. Yeah, we might need to get in early or late or both, which might mean 2 operations 18 months apart, or one tool followed by another tool 15 to 18 months later.
Thanks, Josh. There's a question from John I now. So are there situations where aerial 1080 shouldn't be used at all, and we're left with, and in which case we're left with no small bird protection tools there?
Well, I don't know. I mean, I can't necessarily think of situations where it shouldn't be used at all, other than for socio-political type reasons. I think eradication operations with 1080 are potentially potentially fraught, but still nonetheless might occasionally work. Yeah, be interested to know if anyone else can think of any examples where it shouldn't be used.
There's another question here from Anonymous. Do you have any thoughts on what this means for open, non-fenced mainland islands?
Well, yeah, I think for open non-fenced mainland islands, depending on where you are, I think we've seen some attempts to control growing rodent populations in open non-fenced beech forests at Rotoiti, where operation and in the Dart Valley down south, where ground-based bait station baiting has been unsuccessful in the presence of lots of seed. And so I think that, in those cases, just working out, how to avoid those growth phases is the challenge. In the warmer North Island ratty forests, I think we know that you're going to have to come back quite a lot, and I think this data set's just telling us that we need to keep a varied toolbox going. You're going to have to come back quite often, and you're going to have to use everything that's there. There's still some other aerial tools that we don't tend to use these days that are available to us, and we don't really have. We're not in the habit of using them, and we're losing the kind of technical skills you need to, or hardware to do it, such as the aerial pindone operations and aerial 1080 carrot operations, which I think we should be keeping in our toolbox just to keep our standard operations working for longer.
And we can probably squeeze in one more, Josh. So another one, anonymous one. How do you think resistance is working if, given the life expectancy of rats, very few rats would endure a second aerial op, and certainly not a third?
Yes, yeah. Well, I think given the intervals between these operations that are within this data set, I don't think bait shyness is a big part of it. And in fact, in our analysis, we actually put aside one exceptional operation that was only 6 weeks after the first one, because we're thinking this is largely not about learned aversion from rats surviving between operations, but actually quite likely to be selection for genes that enable survival. And this could be either through behavioral mechanisms. You're selecting for just enhanced caution, more stringent testing of novel foods, or just plain aversion to something like eating cereal foods, which has actually has happened overseas, believe it or not, with rodents, with mice. And then on top of that, finally, of course, there's potentially development of physiological tolerance to high levels of 1080 in your system. So there's a whole raft of possible kind of pathways to populations resisting it, those controls, and we just keep doing it with cereal cinnamon 1080 pellets.
Okay, thanks, Josh.